In order to determine blood type, we will visually centrifuge these blood samples to separate the blood components. The ABO blood group is based on the presence or absence of two major proteins in red blood cell membranes, agglutinogen A and agglutinogen B. A person can have A and B, or neither A nor B in his or her blood cell membrane. These blood types are referred to as A, B, AB, and O. If a person has type A blood, his or her blood will contain antibodies against agglutinogen B. These antibodies are called agglutinins. Conversely, a type B person will have agglutinins against agglutinogen A. Blood compatibility can be predicted by understanding the differences between the donor and recipient blood types. The donor's blood generally only contains red blood cells with no plasma. Therefore, it is agglutinins in the recipient's blood plasma that determine if the donor's blood will be compatible. For these reasons, type O is known as the universal donor, while type AB is known as the universal recipient. The heart operates as two parallel pumps. The right side of the heart circulates blood through the lungs. The left circulates blood through the other parts of the body. The heart's contractile activity results in pressure changes that propel blood through the circulatory system. These pressure changes occur in a cyclic nature and are referred to as the cardiac cycle. The conduction system transmits electrical impulses via specialized cardiac fibers, ensuring that the heart muscle contracts in a coordinated fashion. During the cardiac cycle, specific electrical events occur that can be picked up by electrodes placed on the surface of the body. An electrocardiogram is a graphic display of the heart's electrical activity. The closing of the valves in the heart produces sounds that are often described as lubbed up. The lub is produced by the closing of the AV valves. The dub is produced by the closing of the semilunar valves. Because the heart must continually beat, the coronary arteries serve a critical role, supplying the constantly active heart muscle with oxygenated blood. As we zoom in to observe the interior of this diseased coronary artery, notice the partially restricted blood flow due to the atherosclerotic plaque in the arterial wall. If part of this plaque ruptures, a thrombus forms and may grow and occlude the vessel. The thrombus detaches, causing an embolism, obstructing the blood flow through the artery. Since the supply of blood has been obstructed from reaching the region of the heart supplied by this artery, the myocardial cells become ischemic, resulting in damage to the heart muscle. Symptoms of a myocardial infarction may include shortness of breath, tightness in the chest, intense prolonged chest pain, nausea, fainting, and intense sweating, pain in the left shoulder, arm, jaw, and back. A portal system contains two capillary beds in series. Blood flows from the heart through the first capillary bed, where it then drains into a portal vein. The portal vein carries the blood to a second capillary bed that drains into a vein that returns blood to the heart. The hepatic portal system is one example of a portal system. Its first capillary bed is located in the intestines and the second in the liver. Lymphatic vessels recycle excess tissue fluid that accumulates in the interstitium as a result of normal capillary leakage. The walls of the lymphatic vessels act as one-way valves, only letting fluid enter. Once in the vessels, the lymph is advanced toward the systemic circulation. Along the way, the lymph is filtered through several lymph nodes where it is inspected for foreign substances. Upon reaching the thoracic duct, the lymph re-enters circulation. Phagocytic cells are specialized to non-specifically consume and digest antigens. The macrophage is an example of a phagocytic cell. In addition to this non-specific cleanup function, macrophages digest engulfed antigens and place small antigenic pieces on their surface for presentation to lymphocytes. Fever is a non-specific immune response to infection. Bacteria entering the body through a cut may cause an infection. Upon recognition of the bacterial invasion, lymphocytes release pyrogens that signal the brain to raise body temperature. An elevated body temperature creates an unfavorable environment for microbial growth. A fever also enhances the normal immune response.
Part of nonspecific immunity is a set of about 20 proteins called complement. One mode of action of complement proteins is to form a membrane attack complex when triggered by foreign cells or body cells infected by virus. Clusters of complement protein perforate the cell, whose contents leak out. Complement also stimulates phagocytic white blood cells to engulf these foreign or infected cells.
When a cell is invaded by a virus, one of the mechanisms for a non-specific defense is to protect surrounding cells from infection. Upon viral infection, interferon may be released from the infected cell. This initiates antiviral defense mechanisms in all cells that receive the signal. Although the virus replicates and the infected cell dies from the viral attack, protection of the surrounding cells helps avoid a widespread infection.
Antibodies are proteins that play an integral role in the complex system of specific immunity. They bind to and inactivate foreign proteins called antigens. Each type of antibody binds to one kind of antigen molecule. B cells are one of the two types of white blood cells that make antibodies. When a bacterial cell enters the bloodstream, proteins on its surface act as antigens and trigger response by the immune system. These antigens will bind to the unique B cells that have the corresponding antibodies on their surfaces. These B cells now divide. Some continue to divide, producing many plasma cells. Plasma cells secrete large quantities of antibodies into the bloodstream. This division of only the appropriate B cells is called clonal selection. There are a number of types of T cells involved in specific immunity. T cells recognize their specific foreign protein or antigen only when it is presented on the surface of a body cell. One type of T cell is the cytotoxic or killer T cell. Its role is to destroy infected or cancerous body cells. In this case, a pathogen enters the cell on the left, and that cell moves foreign protein to its surface. A patrolling killer T cell recognizes and binds to that protein and releases perforin, a protein that ruptures the membrane of the infected cell. A second type of T cell is the helper T cell. One of the roles of a helper T cell is to activate B cells. At the top of the screen, a phagocytic white blood cell is presenting antigen from a pathogen it has engulfed. The B cell that responds has antibodies for the same antigen that activates the helper T cell. The T cell sends out chemical signals that affect various immune cells. These chemicals stimulate the B cell to divide and produce a clone of antibody-releasing cells.
Vaccination is the process of exposing an individual to an antigen to stimulate an immune response and produce antibodies to that antigen. Vaccines employ the benefits of priming the immune system for exposure to a pathogen. Many vaccines are attenuated or disabled forms of a pathogen. These attenuated pathogens are recognized by lymphocytes, resulting in clonal expansion and production of plasma cells and memory cells. The memory cells lie dormant until later exposure to the virulent pathogen, at which point they become activated. This secondary response will involve contact with more immunospecific memory cells initially, which results in a faster, more robust immune response.